video in my Beginner's Guide to Budgerigar Genetics series, where we're looking at the colour mutations and varieties in Budgerigars. In this video, we're going to continue to explore those genes that are connected with particular chromosomes. This follows on from the last video where we looked at the genes that were connected to the sex chromosome. If you haven't watched that video or need refreshing on your understanding of how that particular chromosomes work, please do follow the link now and then come back to this video. So before we delve in and explore those genes that are chromosome linked, let's just have a quick refresh on our understanding of Budgerigar genetics. We know that the genes are located in a particular place on a number of chromosomes, and that these chromosomes are held within the cells of each Budgerigar. And there are two pairs of chromosomes, one inherited from the cockbird and one inherited from the henbird. We know that when a gene mutates, it can produce a different variety or a different colour, as in the case of the mutated gene that produces the blue bird, or the blue series birds. We know that when there is a mutation, the bird can be of one of three particular types, and these are called their genotypes, and that is, it can have two of the non-mutated genes, two of the mutated genes, both of these are known as homozygous, or one mutated gene and one of the non-mutated gene, and this is known as heterozygous. We also know that genes can be, or the mutated genes can be either dominant or recessive, and that means that it will either look like the mutated gene or look like the non-mutated gene. We also know that there are some genes that are co-dominant, in which case there's an in-between stage. So in a dominant and recessive type variety, you either look like the particular mutation or you don't, in terms of the phenotype. But in the co-dominant or type varieties, there is an intermediate stage. So there are three varieties that it can look like, or three phenotypes. So one for each of the genotypes. Right, now we've explored that, we went on to look at, in the last video, those varieties that were associated with a particular gene, and that, a particular chromosome, and that chromosome was the sex chromosome. And we also know from that last video that there are a number of varieties that are associated with the sex chromosome, so such as the Ino, the cinnamon and the opaline varieties are all sex-linked and all associated with the sex chromosome. This must mean that all of those gene types are on that particular chromosome. So, if that's the case for the sex chromosome, then must be the case for other chromosomes. And indeed that is the case. So what we will do now is look at the implications of that in terms of our Punnett square, in terms of our prediction. And we'll start off with looking at the dark factor birds that we know exist alongside the same chromosome as that produces the colour series, so the blue gene. Let's go over to the computer and explore what differences that makes in terms of our outcome prediction for a particular bird.
Hello, and here we are back at the computer with the usual Punnett square, which you should all know how to use. If you don't, then please watch the previous videos. We're going to look here at the way we would do a dark green split blue with a sky blue bird in the manner that you've seen previously, where we've just carried each of the genes over as before. So the dark green split blue contains one of the mutated dark green genes and one of the non-mutated dark factor genes, and then contains one of the non-mutated green genes and one of the mutated blue gene, and therefore it can pass on 4 possible combinations. So it can pass on the mutated dark factor and the normal green gene, it can pass on the mutated dark factor and the mutated blue gene, or it can pass on the non-mutated dark gene and the normal green gene, and the non-mutated dark factor gene and the blue gene. The sky blue, of course, can only really pass on its blue gene. I put the non-mutated dark factor gene in there just to even the process up so you can see it. And as you can see from there, there's the various outcomes. So they're the same both top and bottom in terms of the sky blue. So we would end up with a dark green split blue, 25%. We would end up with 25% cobalt. We would end up with 25% light green split blue, and we would end up with 25% sky blue. However, that doesn't take account that the dark factor gene is sat on the same chromosome as the gene for the colour mutation. So this means that when it passes its chromosome over, it would have to pass it over complete. So let's have a quick look at what the actual expected outcome would be based on that particular understanding. So if I click on the next one, we can see here, so we've got the same thing here. We've got a dark green split blue bird crossed with a sky blue bird, but in this case it can only pass on the chromosome with the two genes connected. So in this case we've looked at it and we've said that the dark green gene in this top square up here, the one I've just highlighted, so the dark factor gene and the green gene are passed on the same chromosome, so it will only pass those on as a single mutation, and the non-mutated dark factor gene and the blue gene on the same chromosome, so it can only pass those on in the same process. So in this case, I mean, clearly the sky blue is exactly the same, so in this case we're going to end up with 50% dark green birds and 50% sky blue birds. So completely different outcome than you might have expected had you just carried out the normal one. So the one back here that we just saw, so where we expected 25% of each of those colours, what we actually end up with, because of the way the genes are on the same chromosome, what we actually should end up with is 50% dark green split blue and 50% sky blue. So there'll be no dark factor blues. So in this case here we've looked at, you'll notice that in terms of our dark green split blue bird, that we've said that the dark factor gene linkage is on the green, the chromosome that contains the non-mutated green gene. So what would happen, so let's have a look at what now is to just introduce you to the various types of the dark green birds that we could have and how we get the two different types. So over here on the left hand side we've got an olive green that's being crossed with a sky blue. The olive green clearly contains two of the mutated dark factors, that's what's making it olive green rather than dark green, and the sky blue contains two of the mutated blue genes but none of the mutated dark factor genes, and therefore it's going to be sky blue. The outcome of that is 100% dark green birds, and those are dark green birds similar to the one that we looked at in the previous crossing, in that the dark factor remains on the green the chromosome that also contains the non-mutated green gene, and the non-dark factor it sits on the chromosome that contains the mutated blue gene. So you can see how the inheritances work there and how the two chromosomes have stayed separate in terms of the way they can then be passed on. Over on this side over here we've got a mauve bird, which is contains two of the mutated blue genes. That's what makes it blue series, and therefore it also contains two mutated dark factor genes, and that's what makes it a mauve bird. The light bird is merely the wild type genes, so it contains the non-mutated green and none of the mutated dark factors. And in this case, you can see it's the opposite way round, in that the dark factor now sits on the chromosome that contains the mutated blue gene in all the cases, although the bird's phenotype, they look exactly the same. So the phenotype of these two birds are both dark green, but actually their genotype, we now know, is going to be different in the way the chromosomes are marked, or are housed. So in theory they are two different genotypes, although the phenotype is exactly the same. But we need to now distinguish between these two types in terms of where the dark green mutation, which of the two chromosomes it sits on. So in this case over here, where the dark green or the dark factor sits on the same chromosome that contains the gene, the green gene, a non-mutated green gene, we call that a type 1 bird. And the one over here where the dark factor sits on the mutated blue gene, we call that a type 2 bird. So those are the two possible outcomes in terms of the crossings and the way that the dark green bird can pass on, or in theory can pass on, the various dark factor are linked to the chromosome. So there are two types: type 1 where the dark factor sits on the same chromosome as the non-mutated green, and type 2 where the dark factor sits on the mutated blue gene. If you'd like, what you'd like to do now, if you feel you want to, is have a look at pairing this bird across in the same way as we did here. So pair that bird in the same way here, so using a dark green split blue, but with the chromosome sitting on the blue, with the dark factor sitting on the same chromosome as the blue. So have a look at that. You can do your own Punnett square and work out the outcomes for yourself. So whilst you're doing that, I shall go back and have a back to the birds, and we shall discuss this a little bit further.
Well, I hope you found that interesting and you managed to follow it and understand it. But it does produce something quite interesting, and let's have a look at our lacewing that we started with. Well, we know that the lacewing variety is actually a mixture of two different varieties, and that is the cinnamon mutation and the Ino mutation brought together on a single bird. But we also know that both those mutations are contained on the same chromosome, so the sex chromosome. So even if we had a cockbird that was contained a cinnamon gene on one of its chromosomes and the Ino gene on the other chromosome, whenever it was bred with a normal hen, it would only ever produce either cinnamon hens or Ino hens if we follow the normal process of it passing on the chromosome in its entirety, a single chromosome in its entirety. But we know that we do get lacewing birds. In fact, we've seen one, and here's one here now. So how do we manage to get the lacewing variety when the chromosome is contains only one of the particular mutations? Well, what we do need to start off with is a bird that contains both of the mutations. So a cockbird that contains both the cinnamon mutation and the Ino mutation. And it's all down to the fact that chromosomes are not particularly stable. And we know that the genes are, in fact, rather than being on a single chromosome and just the bands as we saw in the original diagram, we know that the genes are actually more like bubbles, so they actually join to make the chain that is the chromosome. And then occasionally these chromosomes can split, and that they then get muddled up and then reform with the second of the two chromosomes. So I'll just go over to the computer and explain that now in a bit more detail using the diagrams, and I'll see you back here in a bit.
Hello, and I hope in this diagram will explain a little bit about how the the mutations can jump from one chromosome to the other. So if we take a look on the left hand side here, so the area where we've got a pair of chromosomes, and I've tried to highlight the various mutations. So on the left hand side we've got the bird that we looked at earlier, the type 1 dark green bird with the dark mutation. We can see it at the bottom end marked with a D gene on the same chromosome as the non-mutated green gene, and on the right hand side we've got the mutated green gene, so the blue mutation on the non-mutated dark factor gene. So there's our dark green split blue with the type 1 bird. Well, I said earlier that the genes can be fairly unstable, and they often can, or they can become tangled, and they then split and then rejoin. So here we go. So we can see that the tangling's taken place here, and when they rejoin, they rejoin slightly different. So the rejoin and the two chromosomes join and in different sections. So in this case here we can see that the mutation would have moved from the green, the chromosome that contains the standard green gene onto the chromosome that contains the mutated blue gene. Now we do know that this happens because anybody that has bred a type 1 dark green will know that they do occasionally get cobalts out of the pairing. So anybody that had seen the earlier crossing would have gone, ha, that's not true, because actually this, and I'll just quickly click back to it. So this one here, so would have looked at that and gone, actually I know when I pair that I actually do get the occasional cobalt here. So we do occasionally get one of these birds turn up, and in fact this here is how that happens, because occasionally the chromosomes become muddled, they split, rejoin, and that enables the mutation to cross over, and that's exactly what happens in terms of the lacewing. So occasionally a bird that contains a cockbird that contains both the ino gene and the cinnamon gene on two different chromosomes will occasionally, those chromosomes will split and they will cross over. So how do we predict the likely outcome? Well, that's very, very difficult. To some extent, the level of outcome in which this can be produced, and the number of times it happens, is linked to how far away the two alleles, or the two locuses, in terms of on the gene. So here we can see, this is just a representation, but I've placed the two a fairly long distance away. That means that if it splits there, or anywhere up here, then actually when it rejoins they will be on different ones. The closer they are together, the least likely they are that when they split and rejoin they'll be on different chromosomes, because the likelihood is that where the split happens, it will be still leaving the two genes on the same segment of the chromosome. So in terms of the dark factor, we know that it's a fair distance away from the colour mutation because it happens reasonably often, whereas we know that the ino gene and the cinnamon gene are actually quite close together because it happens much less often. In fact, the chances of it happening are about 3%, so you'd only get it happen 3 times out of every pairing. So that's why it's a less common mutation than the cobalt would happen in terms of that pairing. I do hope that explains how the genes can jump from one to the other, and again makes pairing budgerigars and trying to predict the outcome even more interesting. What I'll do now is we'll go back over to the birds and finish off.
So there we are. That's about all we've got time for in this video. We now can see how we would predict outcomes for birds that have the mutation on it, or 2 mutations on the same chromosome. And we also know that chromosomes can be strange things and can always throw up something completely unexpected in the nest, even though we've made some sensible predictions using the Punnett chart. And that makes breeding budgerigars and the various varieties even more interesting. And even when we do understand how a particular pairing should produce in terms of its offspring, there's always that odd chance that those chromosomes will get mixed. So anyway, I hope you enjoyed that video, and I'll see you again next month.